This is the third final part of the motor systems lectures, brain input to the motor systems. So we're gonna start talking about the inputs to area 6. We finished talking in the previous lecture about area 6 as that area that provides kind of the final word before the motor cortex gets activated. Now, the area 6 itself receives a lot of input from the thalamus, which isn't at all surprising, actually. So what is happening? What kind of inputs are we talking about? The major input to this area is actually provided by the basal ganglia. I want you to recall our discussion about sensory pathways, and I want you to recall the different nuclei of the thalamus that we had talked about. So thalamus is this major relay station where a lot of sensory and other inputs are actually processed and then returned back into the cortex. And so this is what happens in the VL, ventrolateral VL nucleus of the thalamus. The VL, sometimes it's called VLO. This is the major motor nucleus of the thalamus. And this is going to be the part that's influenced by the basal ganglia. Basal ganglia is, as the name implies, formation that's deep in the telencephalon, containing lots of white matter, axonal tracts, and some gray matter, which will be the cell bodies in here. So the parts that are interesting to us are the striatum, which is right here. The striatum consists of two bits, the caudate nucleus and the putamen. Okay, so these are the major parts of that we're going to talk about. And then the right next to it, there is something called the globus pallidus, or the pallidium, and there is an external and internal part, and these two will have different functions. And finally, other players here are going to be the substantia nigra, which is this dark substance. The name comes from the dark coloration of the neurons in here, and this is going to provide major modulatory input to the basal ganglia. So let's look at the function here. Let's look at what is happening. Here is another picture, a diagrammatic picture of the basal ganglia, so we can see how deep their location is, and a schematic version of the globus pallidus. And then you can actually appreciate that this portion of basal ganglia is positioned right above the hypothalamus, Now, how were all these experiments done to actually figure out that the basal ganglia is involved in the direct input into the motor pathways? So actually, a lot of these experiments were done in decorticate animals. Decorticate animals are animals where the cortex has been removed. If you remove the cortex, what are you left with? What you're left with is actually the thalamus, the basal ganglia, and the hypothalamus. And you might venture a guess that such animals would die, but that's not the case. These animals can actually live. They can move around, they can eat, they can drink, and they can execute pretty sophisticated motor patterns. If their hypothalamic nuclei are stimulated, they will actually be able to execute movement looking for water. They'll position themselves to drink, and they will drink. So where is this behavior coming from? This behavior is coming from mainly the motor programs. A lot of them are going to be encoded in a spinal cord, but also there's going to be an input of the basal ganglia in here. Okay, so This is a three-dimensional picture of what the basal ganglia actually look like. It's a three-dimensional structure, and here is the amygdala. We'll talk about amygdala later, but maybe it's a good time to actually learn the location. We'll talk about amygdala when we talk about emotions and fear. Here's the caudate nucleus, and you can see that actually winds around. And the globus pallidus, it's a quite large formation here deep in the brain. And the putamen is sort of on the outside of the globus pallidus. The globus pallidus is the internal part here. Okay. Here is a schematic of what the basal ganglia do in this large schematic that you're already familiar with, that contains our spinal pathways and also our motor cortex pathways. So we've already talked about the role of area 6 and area 4, which is the M1, and then the M1 feeding into the corticospinal tract, the rubrospinal pathways, and the ventromedial pathways. We will eventually fill in this gap when we talk about cerebellum in just a moment, but for now let's look at the basal ganglia loop here. So what you can see immediately from this schematic, that the input to basal ganglia is actually provided by the cortex. So it is a loop. The information comes from the cortex, goes into the basal ganglia, gets processed, gets returned into VLO, which is the ventrolateral nucleus of the thalamus, and then it's fed back directly into area 6. And then you already know that from area 6, the information is going to promote direct activation of the M1, which is area 4, the motor cortex 1, and then the action of the muscles downstream here. So this is why the basal ganglia is actually a loop. How does this loop work? It gets a little bit complicated here. Now, I wanted to put up this slide to sort of show you what you do and do not need to know. So this is a fairly complex schematic of how the communication works here, and I actually don't think your book does a great job explaining it. So I have provided you with a couple more resources to help you study. Those are links to 2 different sites. In particular, check out the McGill University website. That one explains the motor loop very, very easily, and it gives you some really nice schematics. So I would like you to use that to learn. So briefly, let's talk about this motor loop, how the signal here gets propagated. So here, I like this pathway a little bit better. This is a diagrammatic pathway. So we have 2 different pathways through the basal ganglia. We have the direct pathway, and then the direct pathway, it's cerebral cortex directly activating or excitatory input. So pay attention to the color of the arrows here. Excitatory input into the striatum, which again is the putamen and the caudate nucleus together. And then that has a direct effect on the globus pallidus internal. And notice that this is an inhibitory, and then again inhibitory from globus pallidus into the VLO of the thalamus, and then there is an excitatory connection back into the cerebral cortex. And then there is the indirect pathway. The indirect pathway differs because it has an additional input from the substantia nigra. So those are the dark colored neurons here, and this we really won't talk about so much, the subthalamic nucleus that provides additional input into the globus pallidus. But without going into too much detail, what do these things do? So the take-home message here from this is that the output neurons here are inhibitory. Okay, the output neurons here are inhibitory onto the thalamic nucleus. So essentially what this pathway has to do is overcome the inhibition. The basal state, if you will, prior to any activation is inhibitory, okay? This is the thalamic input here. We keep inhibiting the thalamic nucleus. So at resting state, these neurons are going to be tonically active. What does it mean tonically? They're just going to keep firing. When they fire, they inhibit, 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 inhibit. So what do you have to do to actually stop them? Well, you're going to have to provide input through this pathway. And you can notice here that both the direct pathway and the indirect pathway will actually modulate the GPI activity by inhibiting it. So if you inhibit the inhibitor, you, in the end up activating the thalamic nucleus. And that's what really happens here. Now, the second really important part here is that the activation of the striatum is notoriously difficult to achieve. These neurons here are not very easily excited. It might look so on this picture, but they are not. They, in fact, need, for their activation, they need modulation of by dopaminergic system, the diffuse dopaminergic system of the brain, and in particular, the substantia nigra, the dark substance of the brain, substantia nigra dopaminergic neurons. These neurons, these dopaminergic neurons, will need to modulate the striatum in order to activate these neurons. Now, keep that in mind, make a note of it. It's going to become important in just a moment when we talk about diseases of the basal ganglia. Now, if you damage this part of the brain, or you have a lesion in the dopaminergic pathway, what's going to happen is you're going to have a difficulty initiating movement. Okay, you're going to have a great difficulty initiating movement. And the converse condition would be if you no longer have this inhibitory function present here. You can think of this inhibitory function as the globus pallidus inhibitory function. It's something that prevents you from doing unnecessary movement, sort of trims all the shakes and the tremors and the waves and the twitches that your muscles would be doing if they didn't know any better, if there was no precise program. So this is what this part of the pathway really does. If you damage this, what you're going to have is an overactive thalamic input here that is just going to go right back into the cerebral cortex and induce all kinds of strange motor programs in the brain. Okay? Take a moment, try to digest it and maybe read the chapter. It does a reasonable job explaining that part. So see if you can kind of catch up with this, and then let's look at the basal ganglia disorders, which hopefully will make it understanding a little better. The first one is Parkinson's disease. I'm sure you have heard of Parkinson's. It's the second most common disorder of the basal ganglia. What is the first one? Think about it for a second. I hope most of you have guessed. It's Alzheimer's disease. Yes, Alzheimer's disease actually affects the basal ganglia. Now, this is a side note here. Knowing that, you immediately can guess that the basal ganglia has additional role that is very, very important in making and storing memories. So this is an additional role that is essential, not just as in the motor loop. That's not the only function of the basal ganglia. Okay, so keep that in mind. We'll go back to the basal ganglia eventually in one of our discussions. Okay, Parkinson's disease. What happens in Parkinson's is patients who have it have hypokinesia or akinesia. They also experience rigidity and tremors, so shaking of hands and shaking of the jaw. The problems initiating movement, if you go back to this loop right here, come from the fact that the dopaminergic neurons here have been damaged. And in fact, that is what happens in Parkinson's. The substantia nigra neurons get damaged. They no longer can provide enough dopaminergic stimulation, modulation to the pathway. So while the subjects are unable to initiate movement, once they initiate movement, they will continue it, and then they will again have a hard time stopping it. But it is the initiation of willed movements, so the movements that are controlled by the cortex, that's affected here. There are some treatments for Parkinson's disease, but they don't really treat the disease itself, because as far as we know, it's not treatable at this point, since it is a destruction of neurons. The patients can be helped, at least with the symptoms, for some time by delivering L-DOPA to them. So L-DOPA, if you go back, way back, when we talked about precursors of neurotransmitters, L-DOPA is a precursor for dopamine. And the good thing about L-DOPA is that it crosses the blood-brain barrier, meaning gets transported right into the brain, and you can literally feed the remaining SN, substantia nigra, neurons with L-DOPA, so that the ones that are still alive can function a little bit better. They don't have to synthesize their own L-DOPA. It takes less energy, and they can still function a little bit. But you can't really stop the destruction of the remaining neurons. So there are additional symptoms there are additional problems in Parkinson's disease. So it's not just the motor movements, of course, but this is one of the major problems. Now, the converse basal ganglia disorder is Huntington's disease. Huntington's disease is when you lose neurons in number of places, caudate nucleus, putamen, and also the globus pallidus, which is sort of the policeman, inhibitory policeman. So now that you've lost the inhibition, so let's look back here. Okay, here is the GPE and GPI, the two portions of globus pallidus that are inhibitory on the VLO, ventrolateral nucleus of the thalamus. When you lose those, you have chorea or hyperkinesia. These people look like they are dancing at all times. And I had put a short movie on the website, and this is a person who is being affected with Huntington's disease, and you can see the symptoms in the way this person is trying to talk and the way that their face is moving. So they're still in the very early stage of Huntington's disease. Now, it is really horrible disease, and it is actually a genetic disease. The chorea is followed by dementia and personality changes, complete loss of personality, really, and it is a condition that ends in early death. So both of those diseases, Huntington's disease and Parkinson's disease, are the disorders of basal ganglia. Now, all the additional symptoms of this disease show you that there is more to basal ganglia than just the motor pathways. Okay, here is a brain of a person who is normal, and here is a damage of the Huntington's disease. And you can see that the damage is not limited to the basal ganglia. Now that those ventricles are huge and the white matter is missing, you can see that there is a diminishment of the gray area, and there is a destruction of the brain in other areas, which is commonly seen in Huntington's disease. Okay, now we're moving along. There's a couple more topics we're going to touch on and finish this. Remember back when we talked about the coding of sensory pathways and how the neurons actually code? well, in the movement in M1, the neurons code in a way that's somewhat similar to the coding of the olfactory neurons. We have these broadly tuned neurons, and each direction, each movement is encoded by activity of many different neurons, several different neurons. So there is no one-to-one representation like we have, like something like we have in the visual cortex. It's more like a tally. There is a bunch of neurons and they're voting which direction we're moving, and the vote kind of is a democratic vote. The majority gets to choose. This is really the only thing that you need to know about the coding of movement. Your book goes into more details, but I'm not requiring you to learn any of it. Okay, the last portion that we're going to talk about is the cerebellum. Cerebellum is the little brain of our brain. Cerebellum is kind of the missing part in our diagram. I'm going to go to the diagram for a moment. Here we go. This is the diagram that we've been working on, and maybe you cannot see the spinal cord, it's a little bit lower, but again, we've talked about our basal ganglia input loop just a moment ago, and now the final part is the cerebellum. Okay, and the cerebellum, again, we're going to deal with the loop that is going to be get input from the cortex, various areas of the cortex. The input is going to be delivered via pons, because that's where the cerebellum sits, on the cerebral peduncles. So there are these powerful axonal tracts that go into the cerebellum, and then from the cerebellum, it's going to feed back into the thalamus through a second thalamic nucleus called VLC, okay, unlike the VLO. And what is important here, this feedback loop is going to feed right into motor cortex M1 into area 4. Okay, so this is going to be a loop that's important not so much in planning the movement, but in the actual execution, of the movement, the precise program to execute a movement. Let's briefly look at the cerebellum. And here is the anatomy of the little brain. It consists of 2 hemispheres and the vermis. And it's actually structured very much like the cerebral part of the brain, like the cerebrum. So it's got the gray matter and the white matter, and then we have these little folds. These are folia. And then we have these deep cerebellar nuclei here. Okay. Now the function of each part of this is the vermis. So that's the central axial part, the central part of the cerebellum. This part controls the axial musculature. So that's going to be muscles that control our posture and abilities to stand up and to keep balance. So those will feed into the ventromedial pathways. And then the 2 hemispheres are going to contribute to the lateral pathways and control the movement of limbs, so the distal muscles. The cross section, this is a particularly nice staining of the cerebellar layers, and you can see the Purkinje cells that you're already familiar with, and the granule cells here. Okay, the cerebellum itself consists mostly of these tiny little excitatory cells called granule cells. They're really, really numerous. And then the granule cells will synapse onto the fibers that run through the cerebellum, and then eventually these very large Purkinje cells are sort of the places where all the input converges, and the Purkinje cells are all inhibitory cells. The granule cells are all excitatory cells. The Purkinje cells are all inhibitory. So let's look a little bit more. The majority of axons that are actually coming into the cerebellum are coming from the sensory motor cortex. Some of them are coming from area 6 and some of them are coming from area 4. Then the information that is processed in the cerebellum is really important in learning motor programs. So this is where the brain is comparing what was intended to what was intended to happen, what kind of movement you were intending to do versus what actually happened, and sort of trimming and adjusting the movement to make sure that it's smooth, to make sure that it is precise. This is the area where really precise motor programs, such as programs required to write beautifully or to play an instrument, this is where they are learned. How important is cerebellum? Very important for coordinated movement. I want to remind you what our movements are like without a functional cerebellum. There are very few cases of patients who do not have a cerebellum, but I'm sure you have seen people with the cerebellar function affected. Those would be people who have drunk way too much alcohol. And we know that their movements become really clumsy. They're definitely not very well coordinated. They're prone to lose their balance. And in fact, what they have lost is at that point of their alcoholic intoxication is they have lost the ability of the cerebral cells, in particular the Purkinje cells, to execute their inhibitory action on the precision of movements. So they're no longer able to execute the motor programs precisely. And we know that one of the sobriety tests is actually walking in a straight line. Very, very difficult to perform for people who are under an influence of alcohol. Okay, we'll end this lecture here. This recording is a little bit long. Please watch all the additional videos that I provided for you, and we will move on to the next chapter in the next set of videos.